Next up, we have Dr. Kearney, and I haven't actually had the chance to really meet Dr. Kearney yet, but she's basically my hero since she has agreed to make us a KCNH1 mouse. So I can't wait to hear all about the plans, and thank you so much for taking on this project.
And thank you so much for having me. It's really, as Wendy said at the very beginning, it's a partnership between the basic research labs and the families. You really help us move forward and keep us focused on the important stuff. So can everybody see my slide?
Yes, we can.
Okay. Get my laser pointer. And just in full disclosure, this is going to be mostly aspirational because we have just transitioned into making a mouse model for KCNH1. So why do we want to do this? What's the value of having an animal model? Most importantly, it provides us access to a developing or a fully developed brain. So we get the whole tissue. And that allows us to study the impact of these pathogenic variants on biology at multiple levels of analysis. So we can look at individual neurons, like Dr. George was talking about, the iPSC-derived neurons, but we can also go further and look at synapses, circuits, networks, and the entire system. So both the brain and the body working together. In addition, it's a dynamic system where we can look at the evolution of symptoms over time. And we can also look at treatment response over time. If a treatment has a honeymoon period, but then wanes, we can model that in the animals. We can identify biomarkers of disease progression and look at treatment response with those biomarkers. We can hopefully identify and provide additional evidence for therapeutic targets. And we can help prioritize novel candidate drugs for future clinical trials. If there are 5 analogs in development, the animal models can help pick which one to move forward. And importantly, we can also help evaluate safety. So if we look at Dravet syndrome, which is another rare developmental and epileptic encephalopathy that has been on the scene for a longer time. There have been animal models developed, including one in my lab, but there have been zebrafish and mouse models developed in other labs as well. And importantly, there's a very good preclinical to clinical translational track record for seizures and epilepsy. So we can use seizures as a measurement in the animals, and then that translates often pretty well to what happens in clinical situations. So there are a number of different medications that have been in development for Dravet syndrome. They're just shown here in these bar plots, and they're at various stages of development, including 3 that have been approved in the last 4 years. And there are also disease-modifying therapies like ASOs and gene therapies. And for every one of these drugs in the pipeline, there have been animal models that provided some of the evidence to move them into the clinic. So we have been making mouse models for neurodevelopmental channelopathies as part of the Center Without Walls that Dr. George talked about. He's actually my neighbor at Northwestern. And so we take the electrophysiology data and make decisions about which models to make. But essentially, we go from the clinical genetics from patients, identify the mutation, and then we can introduce that mutation to the same position in the mouse gene. And this really works because human and mouse KCNH1, their amino acid sequence, so the letters that make up the protein, are 97% identical between human and mouse. So really, there are very few changes. And then, as I said, we can study the brain, we can study neurons, we can study the whole animal, and we can also take neurons out. We can study those neurons in a slice that lets us study circuits. So it's a versatile system. So one of the things that we have to do is we have to pick which variants to study. We can't make all of the variants because it's an expensive and lengthy process. So what we want to think about is representing major categories of the clinical spectrum, representing major categories of channel dysfunction, and that's where that high throughput electrophysiology data can help us group things into different types of alterations. For instance, altered gating, where the channel may open too soon. We also, at least initially, tend to pick recurrent variants because that represents a broader spectrum of individuals. And it also gives us more confidence that that particular variant is disease causing. Ideally, there would be an iPSC line available because these are two complementary model systems that can really help inform one another. And then we try to make unique variants that haven't been modeled by other groups. So we know that there's the existing knockout model that Dr. Pardo talked about. And in addition, there's also a knock-in mouse model that was made by Dr. Simons. That's isoleucine 494 valine. So we don't want to duplicate effort and make the same things. So the variant that we decided to go forward with first is R357Q, which is that most recurrent variant that has altered channel gating. And this is the same slide that Dr. George showed that just shows you that this doesn't have, in fact, in the red, have a very profound effect on channel function. So we use CRISPR-Cas9 genome editing. And what that allows us to do is we go into the mouse gene and we just change that one nucleotide from, in this case, from a G to an A. And that is exactly the same variant that happens in the patients. And so we're not making any other additional changes to the genome, hopefully. And so we have done this step so far. We had our first batch of animals born last week. And in another week or two, we'll start the process of molecular validation. So what that means is we go in and check and make sure that we did this and that we also didn't do any other changes. So we screen for predicted off-target sites. CRISPR-Cas9 has sort of predictable off-target effects sometimes. And the way that we've been doing that recently is we just do whole genome resequencing of the founder mice and just make sure that we're not propagating something where we have other issues beyond that single nucleotide variant, because that's all that we want to see. So for animal models, there's kind of there are pillars of model validation. One is construct validity. So is the underlying cause the same? And certainly for a genetic variant, that's pretty straightforward. We're introducing the same variant. So we can check that box. Space validity talks about does the phenotype resemble what you see in patients? And predictive validity is, do you see a similar treatment response? So if we think about face validity, one of the first things that we do when we have a new mouse model is we approach it similar to clinical genetics, and we just examine body morphology. Is there anything noticeable in terms of the body? We can look at various measurements of the skull or the brain or jaw. And this is an example over here of a mouse model that's called koala that has a characteristic craniofacial phenotype, as well as hairy ears. So it's very easy when you're used to looking at mice all the time to see these differences in morphology. We can also look at the digits. So we could look at sort of those distal ends of what in the mice would not be the ends of the paws, not the hands, but there's also a nail bed. So we can look for these characteristic morphological features. The other thing that my lab spends a lot of time doing is EEG. So seizures and epilepsy are an important facet of phenotyping. And see if this video will play. I think it's playing. So this is a mouse with a different potassium channel variant that's having a seizure. And you can see that that's pretty obvious. But we also do EEG, just like you would do clinically, to see an electrographic component of that. And the other thing that we want to look at is the background EEG. And Dr. Gertler talked about this earlier, that you see changes in background EEG in developmental encephalopathies. And this again is from a different potassium channel variant where we can see profound changes in the background EEG. Here we have lots of spiking in changes in the background rhythms. We can also look at various neurobehavioral assays and look at things like learning and memory, anxiety, motor function, social behavior. There's also things for vocalization. And this is really where we rely on families to inform us of what things we should be going and looking at. So then the last piece of the puzzle is predictive validity. So do the drugs that work in patients work in the mice? That's straightforward when you have drugs that work. It's a little bit trickier when you have a rare disease where there aren't really great treatments. So we, in this case, can rely on things where there might be a partial effect, or if there are things that cause worsening, that's certainly something that can help us benchmark predictive validity so that we know that we can move ahead and look for efficacy of novel drugs. So finally, the last thing that we do, and that is a core value of my lab, and I think it's probably the most important thing we do, is we share our resources. So for instance, the Dravet syndrome mouse model, we deposited that at the Jackson lab where any researcher can go ahead and order it. And what that really does is give you a force multiplier effect. So we published that mouse model in 2014. And if we look at cumulative publications over time, so dissemination of the information, all these, the hashed part of the bar are studies that we either initiated ourselves or contributed to. But the filled part of the bar without the hash is studies that use our mouse model, but we had nothing to do with them other than depositing the mice. And so you can see that there's a great acceleration of the research with this sharing. So finally, I just want to acknowledge people in my lab, and particularly Chris Thompson and Al George, who provided me with the R357Q as the front runner for putting into the mice, and the folks in our transgenic core and our funding sources. Thank you.
Thank you so much, Dr. Kearney. We will all be watching with great interest as you pursue the project.